Hi, everybody. Today we are going to talk about enzymes. So previously we learned about energy, and we learned about how energy played a role in chemical reactions, right? There were endothermic, also called endergonic reactions, and then we had exothermic, also called exergonic reactions. So one thing that we went over is we compared the energy of the reactants to the energy of the products to find out if something was endothermic or exothermic. So here we see that energy got absorbed. Here we see that energy was released, and we calculated that by subtracting the end and the beginning. And that total change told us how much energy was released or absorbed. But there's more to a chemical reaction. So first of all, we have to think about this hill that we've got to go over before we can get to the end of a reaction. And notice that it doesn't matter what type of reaction it is, endo or exo, we still have to go over that hill. This hill is activation energy, as you see here, from the beginning to the very top of the hill represents activation energy. And activation energy is the energy that you've got to overcome in order to get that reaction started. So for example, you think about having a piece of paper, right? So this piece of paper could theoretically react with the oxygen around it and combust. But that doesn't happen usually. And the reason why it doesn't happen is because in that reaction, you would have to get over a very high activation energy. So how would you get over that? Well, you would get over by lighting a match and having that match light the paper on fire, which would allow it to react with the oxygen in the air. That would be getting over activation energy. Right, every reaction has a certain amount of activation energy that you have to go over, and this can cause reactions to be very, very slow. Again, it doesn't matter if it's endothermic or exothermic, you've got to get all the way over this hill before you can go back down and finish out the reaction. Okay, so in biology, that's going to be a problem because we need reactions to happen very quickly, and we need them to happen fast, and we need a lot of them. So we have what's called a catalyst. A catalyst is going to lower the activation energy, and we can see that in the picture. The normal activation energy is in blue. It's a very high hill. And with the catalyst, the activation energy is in red. It's much lower and easier to overcome. In biology, the biological catalyst is enzymes. We have enzymes in all of our cells. Every living thing has enzymes. Enzymes are very important. Enzymes are proteins, so that's important to know. They're proteins, and they cause a reaction to go faster. How do they do that? They do that by lowering the activation energy. So now the activation energy is lower, the reaction can proceed very, very quickly right over that shorter hill, rather than the long and slow trying to get over the big hill. Okay, so lecture check, 2 questions here. What is the role of an enzyme, and how is activation energy related to substrate? Take some time to answer these questions now. All right, so the role of an enzyme, it's going to speed up a reaction. How does it do that? By lowering the activation energy. And how is activation energy related to the substrate? Well, we can't get this substrate to react. It has to overcome the activation energy first. So here's a picture of an enzyme. The enzyme is in purple. So there's our enzyme. Enzymes have a very particular shape. Remember, they're proteins. And we already know about proteins, that proteins have a very specific shape. Enzymes are no different. They have a very, very specific shape. It's very important that they have the right shape. And in that shape, there is something called the active site. This is the little part that looks like it's been chewed out. This is where the reaction is going to happen. Here's sucrose. Sucrose is a disaccharide, and sucrose is going to break up into its monosaccharides, which are over here. That could happen on its own, but that would happen very, very slowly. It might take a really long time for sucrose to break up on its own. So we have an enzyme here. This enzyme would be called sucrase, and sucrase is specific to sucrose. We see it fitting in steps 2 and 3. Nothing else can fit in this enzyme. Only sucrose can fit in this enzyme. Other things cannot fit in. The enzyme sucrase is specific to sucrose. The enzyme is going to place a lot of pressure on that bond, and it's going to break sucrose up into its two parts, which are then going to leave. Those are the products. And what we're going to be left with is an empty enzyme, and that empty enzyme can be reused. We can use enzymes hundreds, thousands, millions of times. Enzymes can be used a lot. So the question below, what process is this? So what type of reaction would this be? This would be an exothermic reaction. And you should also be asking yourself, okay, if this is an exothermic reaction, then it also is going to be a hydrolysis reaction because we are breaking up something into smaller pieces. So this represents hydrolysis. Two important things to ask yourself. So some definitions, enzymes. Enzymes are biological catalysts, and they speed up the reaction by lowering the activation energy. You absolutely need to know what an enzyme is. The substrate is the product. It is the thing at the beginning. The substrate eventually is going to react and turn into the product. And then we have the active site, the portion of the enzyme where the substrate binds. Here, it is this part that looks like it's been cut out with a cookie cutter. This is where the reaction happens. All right, here we have a view. Here's our enzyme. There's our active site. Here's our substrate, sucrose. The substrate fits right in. The enzyme is going to apply a lot of pressure to that bond. It's going to break the bond, and fructose and glucose are going to be separated. And these are now your products, and they go on their way. Enzyme is empty, and the enzyme can be used again. It's a constant cycle. This happens very quickly. You could have this happen a lot of times in a split second. So this is all going on very, very fast. And you need this to happen very quickly because this is how we stay alive. We stay alive due to the presence of enzymes. Okay, so what type of reaction did that just show? So two choices there really. Is it a dehydration synthesis reaction or is it a hydrolysis reaction? And then could this have happened without the enzyme? Take a moment and answer those questions. So the answer to the first part, what type of reaction does that show? Previously, you saw a hydrolysis reaction. It was being broken up. That's an exothermic or exergonic reaction. And could this have happened without an enzyme? The answer is yes, but it would have happened much more slowly. Here we have an enzyme doing the opposite. This enzyme, notice it's taking two substrates, and what's happening? It's building them bigger. What type of reaction is that? That's a dehydration synthesis reaction. And here we have two different substrates, and notice at the end, the enzyme has caused a switching of the bonds. So we have a different type of reaction there. So lecture check again. Enzymes can A, break down molecules, B, build bigger molecules, C, both, or D, none of the above. Take a minute and answer this question now. So some other definitions. Enzymes are substrate specific, which means there is a very particular substrate for each enzyme. Like a lock fits into a key. You got the wrong key or the wrong lock, it's not going to work. Same thing, if you have the wrong substrate, it's not going to work with an enzyme. Enzymes are not used up. They can be used over and over again. And enzymes greatly speed up a reaction, and we could not survive without them. All right. So lecture check. Are enzymes used up in a reaction? And what does lock and key refer to? Take a minute and answer these questions now. So your answer, are the enzymes used up? The answer is no, they are not used up. They get used again and again and again. And number two, what does lock and key refer to? It refers to that the enzyme is specific for a type of substrate. All right, another definition between enzyme and substrate, the induced fit model. And this tells us that the enzyme is going to change shape slightly to better fit the substrate. Here we have two substrates and we have an enzyme. They fit in, and as they fit in, what actually happens is, notice the enzyme has changed shape a little bit to better fit those substrate molecules. Kind of like you have a rubber glove and you're putting your hand into it. The rubber glove kind of stretches and morphs around your hand a little bit. The enzyme is doing something very similar here. It then causes these two things to react. In this case, it is synthesizing a bigger molecule, and when they are released, then the active site goes back to its original shape. All right, so in review, chemical reactions are hard to get over, right? We have to get over this entire hill of activation energy before a reaction can happen. Reactions can do that on their own, but it happens very slowly. An enzyme is going to lower that activation energy a lot by providing a place for the reaction to specifically happen, and that makes it much easier for the reaction to go along, and it's going to speed up the reaction. Okay, now a few other things we're going to go over. Enzymes are proteins, and we know that proteins have a very specific shape, and enzymes are no different. If that shape changes, that's a bad thing. If your enzymes change shape in your body, they cannot be used, and those are chemical reactions that can't happen. There are a number of diseases that are caused because our enzymes are not shaped correctly. Just like with proteins, things like heat, salt concentration, sunlight, pH, like in the presence of an acid or a base, these things can denature an enzyme. They can cause an enzyme to become the wrong shape. And if they become the wrong shape, then substrates, like you see this substrate is trying to fit in, but it cannot. If the shape is wrong, then substrates will not fit into the active site and no reaction happens. And that is really, really bad for living things. We need proper enzymes. Okay, so lecture check question. What things can cause an enzyme to denature? Put that in now. So you should have said things like sunlight, salt, heat, acids and bases. Those things would have been good answers to what might cause an enzyme to denature. Finally, the very last thing we're going to go over is we can actually turn on and off enzymes. We don't necessarily want enzymes to work all the time. Some chemical reactions we want certain times, but not all the time. So there are what's called allosteric sites. An allosteric site is a site that looks kind of like an active site, but it's not. This is the active site where the substrate can fit in. But this is the allosteric site where another molecule might fit in. So in this case, we have a molecule made by the cell, and it has fit in the allosteric site. And if you see what has happened, what happened to the active site when you compare? The active site has changed shape, and it no longer fits that substrate. So the substrate cannot fit in anymore. The substrate can't be acted on, there's no reaction. This is a way that the body has of turning off an enzyme when it is not needed. Right? We can actually turn off, let me get my face out of the way. We can turn off an enzyme like you see on the top. The allosteric inhibitor fits into the allosteric site, and that's going to change the shape of the enzyme, and the substrate cannot fit in. We have turned this enzyme off. When we're ready for it, we'll remove the allosteric inhibitor. It's inhibitor because it's inhibiting the reaction. We can also have allosteric activators. They might turn on an enzyme. So here's our allosteric site. This active site is the wrong shape. Notice that is not going to fit in. We add an activator. The activator is going to make the correct shape and activate the enzyme. The substrate can fit right in, and the substrate can react and become product. So we can turn on and off reactions using allosteric inhibitors and activators that fit into a site that is not the active site. So our final lecture check, what is the difference between an allosteric activator and inhibitor? So you should say that an activator is going to turn on an enzyme and an inhibitor is going to turn off an enzyme. All right, and that is where we are going to end today. Those are enzymes. If you have any questions, please let me know, and I will see you later.